Hello friends, welcome to my course on microbial biotechnology. We are in module 10 discussing about microbes in medical biotechnology. Today we will be discussing about the production of vaccines. This lecture is divided into 4 broad sections. In the first section we will be discussing about the basics of vaccine technology. In section 2 we will be discussing about vaccine research. In 3 we will discuss how vaccines are manufactured, and in section 4 we will be discussing some of the advances in vaccine biotechnology. So, a vaccine is a biological preparation which provides active acquired immunity against specific infections or malignant diseases. They generally comprise an agent resembling a disease causing microorganism which stimulates the immune system to recognize, eliminate and prepare to neutralize similar threats in future encounters. Vaccines can be preventive, protecting against future infections, or therapeutic, treating existing diseases such as cancer. Vaccine administration is termed as vaccination, acknowledged as the most potent method for averting infectious diseases. Global immunity, predominantly achieved through vaccination, notably led to the eradication of smallpox worldwide and the containment of ailments like polio, measles, and tetanus in many regions. Let us now look into the history of the vaccine development. Historically, China and Ottoman Empire used variolation to establish immunity against smallpox by exposing individuals to smallpox scabs or fluid. Modern vaccination began in 1796 when Edward Jenner discovered that cowpox infection provided immunity to smallpox. His experiment, inoculating an 8-year-old boy, James Phipp, as you can see in this photograph illustration, with material from a cowpox sore, led to the development of the first vaccine and ultimately the global eradication of smallpox in 1980. In the 19th and 20th centuries, vaccines for rabies, cholera, typhoid and tetanus were developed, forming the foundation of modern immunization. Today, advancements in vaccine technology continue to enhance precision and effectiveness, protecting public health against emerging diseases. So, if you look into the development of vaccine over time, we can divide them into several generations. The first generation basically are referred to as the live attenuated vaccines and also inactivated vaccines. Some examples of the first are the MMR influenza, OPV, chickenpox, yellow fever, hepatitis A, and examples of inactivated vaccines are influenza, hepatitis B, IPV, rabies, cholera, plague and pertussis. The second generation vaccines are called as subunit vaccines. Some examples are hepatitis B, diphtheria, pertussis, anthrax, Haemophilus influenza B, and also some of the recombinant vaccines like hepatitis B, HSV rotavirus, HPV and FMD. The third generation vaccines are basically the DNA RNA vaccines against malaria, HIV, cancers, influenza, Ebola, hepatitis and HPV. Now there is a fourth generation of vaccines coming up. These are also sometimes called as the reverse vaccinology, targeted against AIDS, cancer, dengue fever and malaria. Vaccines stimulate the immune system to recognize and remember specific pathogens such as viruses or bacteria. They contain antigens which are parts of the pathogen that trigger an immune response but are modified to be harmless, either by being weakened, killed or genetically engineered. Upon administration, the immune system identifies these antigens as foreign, prompting the production of antibodies, specialized proteins that neutralize the pathogen. Additionally, immune cells like T cells and B cells are activated, creating a memory of the pathogen's antigens. The primary goal of vaccination is to prepare the immune system to respond quickly and effectively to future exposures, enabling a rapid defense that prevents infection or reduces disease severity. These are general systematics of immune response through vaccination. We will also discuss this in detail with a little bit of more illustrations. The first step is antigens are taken up by antigen presenting cells such as dendritic cells. They are broken down and presented on the surface of APCs using major histocompatibility complex molecules. Then this antigen MHC complex on APCs recognized by helper T cells, leading to its activation and release of cytokines stimulate B cells specific to vaccine antigen. Proliferation of these B cells lead to the activated B cells differentiate into plasma cells which produce and release antibodies, and also activated B cells differentiates into memory B cells capable of rapid immune response on re-exposure in future. Antigens are sometimes broken down and present on the surface of APC using mesohistocompatibility complex MHC molecules, and they directly activates the cytotoxic T cells which recognize and eliminate the infected cells, or they may go through the antigen MHC complex on APC recognized by the helper T cells leading to its activation, and then end up in the same point in the immune system pathways, and these actually generate the memory T cells with ability to induce rapid response on re-exposure. Let us have a little bit of explanation of these systematics of immune response in a little bit more elaborate way, more illustrative way, how a vaccine works when a person is vaccinated. So, the person is vaccinated here, as you can see, this is the skin and underlying the skin is the muscle. So, these are antigens and these are some additives which are called as adjuvants. We will learn about them later. So, these antigens, as you can see, are taken up by antigen presenting cells such as these dendritic cell. So, and then this leads to the activation and trafficking to draining lymph nodes. So, these are broken down and presented on the surface of antigen presenting cells using major histocompatibility complex molecules. So, as you can see over here, these class 1, class 2 major histocompatibility complex molecules are involved in this step. So, these antigen MHC complex on the APCs are recognized by helper T cells. These helper T cells, the CD4 plus marker leading to its activation, and then there is a release of cytokines that stimulates the B cells specific to vaccine antigen. Proliferation of these B cell will then follow. These activated B cells differentiate into plasma cells here, as you can see, which produces and releases the antibodies. That is the number one outcome of this entire effect. Or these activated B cells will differentiate into memory B cells capable of rapid immune responses on re-exposure. Separately, the cytotoxic T cells here having CD8 plus markers, these are activated, these recognize and eliminate the infected cells. So, this step, as we discussed earlier, may also happen directly without going through the cytotoxic T cell pathway. These memory T cells generated with the ability to induce rapid response on re-exposure finally. So, now we know how a vaccine works by different pathways, has different cells involved in it, B cells, T cells, and then memory cells. So, what are the various types of vaccines that are used? We may use a whole pathogen which causes the disease as a vaccine, and these may be further divided into live attenuated vaccines. So, these vaccines use weakened form of the actual pathogen. The potent one or the strong one will actually cause disease. We use a weakened form of that pathogenic entity. These cannot cause disease, but can still stimulate a strong immune response. So, here this is a disease causing virus having some spike protein which acts as antigen. So, this is weakened with different treatments sometimes, and we use it directly. So, these are some of the examples of live attenuated vaccines like measles, mumps, rubella, yellow fever, oral polio vaccines. Then we have inactivated vaccines. These vaccines use pathogens that have been killed or inactivated, so they cannot replicate in the body. So, we may use heat or certain chemicals for destroying the potency, and then these are used as vaccines. Some of the examples include the polio, hepatitis A, rabies, influenza. Then we may have genetic vaccines. For example, we may have DNA vaccines. These vaccines use DNA plasmid containing some viral genes with additional genetic material that will produce some antigenic proteins similar to viral antigens. So, this is a gene for viral antigen, and this is a plasmid which is carrying this particular gene. These are injected into the host, and due to transcription and translation, the antigens are produced within the host. Some of the example include ZyCoV-D. Then we have RNA vaccines. These vaccines use genetic material mRNA that encodes a portion of the pathogen's protein to stimulate an immune response. So, this is the mRNA encoding for the viral antigen over here, and for delivering these we are using a lipid vesicle over here. So, this is injected into the host, and then this mRNA is translated directly into antigen. The difference between a DNA vaccine and RNA vaccine is easily visible from these two illustrations. Some of the examples of RNA vaccines are the Pfizer BioNTech COVID-19 vaccines, Moderna COVID-19 vaccines. So, we may also have subunit vaccines. These vaccines use specific parts of the pathogen such as proteins or sugars without using genetic material. Examples include human papillomavirus, Haemophilus influenza type B conjugate vaccine, or you may have inactivated vaccines. These are inactivated toxins produced by bacteria to induce an immune response against the toxin itself. So, in the earlier case we were using the entire organism, killed it and used it, but here we are only using the particular toxin which is inactivated or denatured. Some examples include the diphtheria or tetanus. So, now we have the viral vector vaccines. These use a modified viral vector to deliver genetic materials from the target pathogen, stimulating an immune response. So, we have a disease causing virus over here. This is a less pathogenic virus. So, here we go for viral vector encoding target antigen. So, these will be finally producing the viral antigen. Then some of the examples are the Oxford AstraZeneca COVID-19 vaccine, Johnson & Johnson's Janssen COVID-19 vaccine. Then we use also virus like particle vaccines. These vaccines mimic the structure of viruses, but lack the genetic material needed for replication. So, some of the examples include human papillomavirus vaccine, hepatitis B vaccine. Let us now discuss in brief about the research in vaccine. Here we will be discussing about the process of vaccine development, and then research and development phase, preclinical trials, and then clinical trials, approval, and finally licensing. So, the process of vaccine research or development includes the discovery phase. The discovery phase of vaccine development involves identifying and characterizing antigens or target components from a pathogen and exploring potential vaccine formulations. This phase lays the foundation for advancing promising candidates for clinical trials. The next phase is the preclinical development. In these, the vaccine candidate is tested in laboratory and animal studies to assess safety, immunogenicity, and efficacy. These studies provide crucial insights into the potential of the vaccine before advancing to human clinical trials. Then next comes the small scale process development, where the delivery system for the vaccine candidate is designed concurrently with preclinical trials while manufacturing is expanded in scale. Protocols adhering to good manufacturing practices are formulated, and the initial GMP level batches are produced, preparing for clinical trials. Then the clinical trial phases starts. In phase 1 clinical trial, vaccine administered to a small number of healthy individuals to evaluate vaccine safety. In phase 2, vaccine administered to a larger group to evaluate immune response and the optimal dose. In phase 3, vaccine administered to about 1,000 people to evaluate protective action against a pathogen. Then comes the evaluation and decision stage. The preclinical and clinical trials are documented, and the vaccine must be approved by concerned authorities such as the WHO, Food and Drug Administration, in the US and Central Drug Standard Control Organization in India. If approved, license to market is issued for the vaccine. Then comes the manufacturing stage. The manufacturing process is designed and optimized as per GMP standards. Vaccine production is done in large scale to ensure that sufficient doses are available. Then there is a safety monitoring clinical trial phase 4 also known as. These are post-marketing vaccines are continuously monitored to assess any side effects reported by the patients or health professionals. So, during the initial phase of vaccine development, researchers investigate their ideas for a potential vaccine. These stages typically spans 10 to 15 years of laboratory research, commonly conducted in private industry with frequent collaboration with university researchers. The primary goals of this phase may be summed up as below. First is the identification of targets. Researchers identify pathogens or antigens associated with significant diseases. Then comes the antigen discovery, where researchers isolate, characterize and study antigens from the target pathogen to identify suitable candidates for vaccine development. Then comes the formulation and design. Scientists design the vaccine considering factors such as the type of vaccine, adjuvants and delivery methods. So, these photographs you can see Dr. E. Hopes and associates working on rubella vaccine in 1965 on the top picture. And then there is the development of COVID-19 vaccine at the National Institute of Allergy and Infectious Diseases recently. So, before a vaccine undergoes human testing, it undergoes preclinical test using lab grown cells and animal models to evaluate the vaccine's safety and efficacy in eliciting a protective response. These tests provide insights into potential human reactions and determine a safe starting dose for human trials. If the vaccine fails to induce the desired immune response at this stage, it typically does not progress further. This phase, often lasting 1 to 2 years, is primarily conducted by private companies. In parallel to preclinical studies, suitable delivery systems for the vaccine testing is also developed. Investigational New Drug IND program helps pharmaceutical companies or researchers to secure permission for initiating human clinical trials prior to the approval of a marketing application. The program is controlled by FDA in the US and CDSCO in India. Researchers are required to provide the details on vaccine manufacturing and testing, summarize laboratory findings and outline the proposed human study. Approval from an ethics committee at the trial institution is essential, and the authoritative organization has a 30-day window for reviewing and approving the application. Upon approval, the vaccine proceeds to 3 phases of human testing, which we have already discussed. The phase 1 basically tests the vaccine's safety and efficacy in inducing immune responses in around 20 to 80 adults, starting with adults before including younger participants for pediatric vaccines. In some cases, volunteers may be exposed to the disease under controlled non-lethal conditions. In phase 2 trials, several hundred participants are involved, including high-risk individuals, to evaluate safety, efficacy, dosage, timing and administration methods. These randomized controlled trials compare vaccine recipients with placebo groups, ensuring groups similarities to minimize individual differences. In phase 3, successful vaccines from phase 2 undergo testing on thousands to tens of thousands of participants, often across different global sites. These trials maintain randomization and double-blind conditions, ensuring that neither participants nor researchers are aware of who receives the actual vaccine or a placebo. The primary goals of phase 3 include identifying rare side effects, evaluating the vaccine's overall effectiveness, and assessing its ability to prevent the disease, halt infection, or stimulate a defensive response in the body. This phase large participant pool is essential for detecting rare events such as side effects occurring in 1 out of every 10,000 people. Then comes the approval and licensing stage. Vaccine licensure follows the successful completion of the development cycle, including clinical trials in phase 1 to 3, demonstration of the safety, immunoactivity, and proven effectiveness in preventing infection for target populations. The World Health Organization Expert Committee on Biological Standardization establishes international guidelines for manufacturing and quality control, providing a framework for national regulatory agencies in the licensing process. Manufacturers receive licensing only after a comprehensive clinical development cycle, scientific review by regulatory bodies such as the Central Drug Standard Control Organization of the Government of India, or the US Food and Drug Administration, and confirmation of safety and long-term effectiveness. In adopting WHO guidelines for vaccine development, individual countries take on the responsibility of issuing national licensure and managing vaccine deployment and monitoring within their borders. Let us now discuss how vaccines are manufactured. So, here we will have a overview of the vaccine manufacture process, then strain selection and growth of the microorganisms, inactivation and attenuation, vaccine formulation, quality assurance, and phase for monitoring. Some of the examples of vaccine manufacturing process from the vaccine type point of view, we may have inactivated vaccine, recombinant vaccine, or live vaccines, which we have discussed earlier in this lecture. So, corresponding to these diseases, hepatitis A, B, or measles, see there are different culture techniques involved. For example, in the case of hepatitis A, which is inactivated vaccine, we have MRC5 human diploid cells. So, it is harvested by lysing the cells to form a suspension, and this is formulated by adsorbing onto aluminum hydroxide. Hepatitis B recombinant vaccine are produced by culturing recombinant cells, and these are harvested by getting them released from yeast to form a suspension, and they are co-precipitate of AGs with amorphous aluminum hydroxyphosphate sulfate as a formulation. Influenza vaccine is an inactivated vaccine which is produced in embryonated chicken egg, and it is obtained by low speed centrifugation and filtration, and it is formulated by using phosphate buffered saline with gelatin as a stabilizer. Measles, mumps, rubella, and varicella are used directly as live forms as vaccines. Measles and mumps are produced in embryonated chicken egg, rubella on WI38 human diploid lung fibroblast, varicella on MRC5 human diploid cells. Various harvesting techniques are used to obtain them depending on the organism, and these are mostly formulated by lyophilizing them into dry powder. Then similarly, we have pneumococcal vaccines, which is basically a conjugate vaccine, and here we use Streptococcus pneumoniae serotypes grown on soy peptone broth, and then Corynebacterium diphtheria is grown on yeast extract based media. These are obtained by polysaccharide isolation by centrifugation, and for formulation we use aluminum hydroxide suspension. Polio is an inactivated vaccine. These are cultivated in Vero cells and obtained by clarification and concentration, and these are formulated in medium M199. Then we have rabies, which is again inactivated, produced on sick and fibroblast. These are obtained by inactivation with beta propionectin. For formulation, these are stabilized with buffered polygellin and potassium glutamate in lyophilized form. So, let us look into the general outline of vaccine manufacture, which is having an upstream processing phase, where we select the antigen or the strain for vaccine production. Then we grow the microorganism. Then in the downstream processing step, we isolate and purify the microorganism. Then we go for inactivation of the organism, and then finally formulation of the vaccine, and after that we go check the quality, and that particular lot is ready to be released if it passed the QA step. So, in the strain selection phase, antigen selection for vaccine manufacturing is a critical step that involves choosing the most appropriate and immunogenic components of a pathogen to induce a protective immune response. Key factors influencing the selection of antigens for vaccine development are: number one, immunogenicity. The selected antigen must elicit a robust immune response. Factors such as antigenicity, antigen presentation and the ability to induce both humoral and cellular immune responses are crucial. Then antigens should be highly conserved among different strains of a pathogen are preferred to ensure broad protection against various variants. The selected antigens should not cause adverse reactions or undesirable side effects. So, safety is an important aspect. Then the antigens that are stable and can be produced in large quantities with consistent quality are preferred for vaccine manufacturing. So, stability is very much important for production. Then considering the age group or target group, the health status and the specific characteristics of the target population is essential for effective vaccine design. So, we should always keep in mind the target population whenever we are designing a vaccine. Once the organism of choice has been obtained from the seed lot, the organisms must be grown such that the desired quantity of organism required to fulfill the vaccine requirement may be reached. Depending on the pathogen, the growth of microorganisms may be studied under two broad heads. Number one, the bacterial growth, secondly, the viral growth. So, earlier we have discussed in module 5, bacterial cultures can be maintained in a batch culture or a continuous culture, and thus bacterial pathogen may be grown in a fermenter, maintaining optimal conditions for growth with the required nutrients provided. In the case of viral growth, cultivating viruses to produce viral materials necessary for vaccine production involves introducing virus into a suitable host cell culture, providing an environment that allows the virus to replicate and multiply. Most vaccine producers use either different cell lines or chicken embryo for virus cultures. Live animals have sometimes been used for vaccine production. Once inoculated, the host is placed in a controlled environment, typically an incubator, where temperature, humidity and other conditions are optimized for viral replication. Once a sufficient amount of virus has been produced within the host cells, the culture is harvested. After harvesting, the viral particles are purified and quantified. Now, let us look into the production of viral particles where we are using egg-based culture systems for vaccine production. Egg-based vaccines require inoculation of fertilized chicken eggs with viral strains, whereby the viruses replicate utilizing the cellular machinery of the developing chick embryo. And we have the cell-based vaccine production as an alternative. The cell-based vaccine production utilizes lab cultured cells for production of vaccines. Cell-based systems enable quicker adaptation to emerging viruses such as novel influenza strains and eliminates concerns related to egg-related allergic reactions. It also particularly useful for viruses that do not grow well in eggs. Then comes the inactivation step. Virus inactivation involves dismantling a virus ability to infect cells without actually eliminating the virus. Virus inactivation works by one of the following two mechanisms: by attacking the viral envelope or capsid and destroying its ability to infect cells, by disrupting the viral DNA or RNA and preventing replication. Some methods of achieving virus inactivation are as follows. We may use solvent detergent for inactivation. Then we use pasteurization or we use acidic pH for obtaining inactivation, particularly low pH treatment, or we may also use ultraviolet radiation for the inactivation, and we may use some certain chemicals for the inactivation. Attenuation is an important process which is used in the development of vaccines to reduce the virulence or pathogenicity of a microorganism while retaining its ability to stimulate an immune response. The goal of attenuation is to create a weakened form of the pathogen that is safe for use in vaccines, but still elicits a robust and protective immune response. Some methods for achieving attenuation are attenuation by serial passage, attenuation by temperature adaptation, attenuation through genetic modification. Attenuated vaccines elicit a more robust and long-lasting immune response with a rapid onset of immunity when compared to inactivated vaccines. So, let us look into some of these methods of attenuation, for example, by serial passaging. This process involves repeatedly passing a pathogen through hosts or cell cultures under controlled conditions, prompting genetic changes. Over time, mutations reduce the pathogen's virulence while preserving its ability to trigger an immune response. Or we may alternatively go for attenuation by temperature adaptation, which involves gradually lowering the microorganisms' growth temperatures to select strains that thrive in cooler conditions. The final attenuated strain replicates well at lower temperatures, but has reduced virulence at the host's body temperature. We may also obtain attenuation by genetic modification, whereby organisms are exposed to mutagens and the mutants with less virulence are selected. Then comes the vaccine formulation stage, where careful design and preparation of the components that make up a vaccine to ensure its safety, stability and effectiveness comes into play. So, here we have these antigen, we use stabilizers, we use adjuvants, preservatives, then surfactants, also buffers, then we may have residual components and diluents. So, these are the various components in vaccine formulation. So, the key components are ones which we have already shown in this illustration. The immunogen, although the active ingredient is present in a minimal quantity with a single dose containing only minute amounts. The adjuvants enhance a vaccine's effects by mimicking pathogen associated molecular patterns to stimulate a stronger immune response against a specific disease. By activating dendritic cells, lymphocytes and macrophages, adjuvants amplify the innate immune response, simulating a natural infection. Some commonly used adjuvants are inorganic compounds like potassium alum, aluminum hydroxide, aluminum phosphate, calcium phosphate hydroxide. Then we may use oils like paraffin oil and squalene. Then certain bacterial products, particularly inactivated bacteria from species toxoids, monophosphorylated lipid A. Then we have plant saponins derived from Quilea, bark extract from certain trees, or soybean. Then we have certain cytokines like IL-1, 2 and 12. Then we have also CpG oligonucleotides, which are used as adjuvants. Then we may have combinations, Frouin's complete adjuvant on Frouin's incomplete adjuvant, AS01 matrix M and so on. Quality assurance, again quality control is very important in vaccine production. It ensures vaccine safety, potency, purity and consistency throughout development, manufacturing and distribution. It includes rigorous testing, detailed documentation of results and deviations and post distribution surveillance to monitor safety and effectiveness. Traceability systems track vaccine batches, enabling swift recalls if needed. Specific tests such as virulence tests for live vaccines ensure they do not retain or revert to virulence, reducing risks to animals and the environment. Environmental risk assessments evaluate vaccine setting, spread and persistence. Stability tests determine the validity of the product's expiry date, ensuring its reliability over time. Then one important phase is the phase 4 monitoring. Phase 4 of clinical trials, also known as post-marketing surveillance phase, focuses on monitoring the long-term safety and effectiveness of the vaccine in a larger and more diverse population over an extended period. Continuous assessments of the vaccine safety profile over an extended period is a primary objective, including monitoring for unexpected side effects, adverse events or interactions with other medications. Phase 4 trials assess how well the vaccine performs under real world conditions, considering factors such as different patients populations, diverse healthcare settings and variations in adherence to treatment regimens. Findings from phase 4 trials may lead to updates in the product's labeling to include new safety information or changes in recommended usage. Let us now discuss about the advances in vaccine biotechnology. We will have an overview of the vaccine biotechnology scenario, then discuss a little bit about the recombinant protein vaccines, DNA vaccines, mRNA vaccines and viral vector vaccines. Research for vaccines that are safer in a broader sense and can also induce a more robust immune response have led to the recognition of genetic vaccines, which utilizes DNA or RNA molecules as potential vaccine candidates. Although the possibility of using genetic material has been demonstrated time and over by several researchers, including Ulmer and colleagues in 1993, Martinon, Mullian and colleagues, and by Zhao and colleagues in 1994, and then the first RNA and DNA based vaccines approved for humans were the COVID vaccines recently. Another type of vaccine that is now being employed are the viral vector vaccines, first used by Moss and colleagues in 1995. Recombinant protein vaccines or recombinant subunit vaccines use antigenic proteins subunits from pathogens produced via recombinant DNA technology. Since these vaccines lack live viral particles, they significantly reduce the risk of infection, making them particularly safe for immunocompromised individuals. When administered, the antigens trigger immune responses, leading to the production of specific antibodies and the development of immunological memory. This memory enables rapid and effective pathogen elimination upon further exposure. Let us look into the principles involved in this case. So, development of recombinant subunit vaccine starts with the identification of the antigens from the pathogen that can induce similar immune response to that produced by the whole antigen. So, we identify this, and this is the antigen gene. Then we have a vector with a selectable marker and multiple cloning site and a promoter to which it will be the antigenic gene will be cloned. Once identified, the gene coding for these protein is selected, cloned and expressed using different vectors in a manner similar to production on any other recombinant DNA product. So, here the antigen gene cloned next to the promoter in the MCS. So, once the protein has been and these are transformed into the host cells, and transcription and translation will follow, which will give rise to the protein. Once the protein has been produced in a suitable host, suitable formulations are developed for the delivery of the vaccine into the patient's body. So, we may use fermenters for scaling up, and finally, these are isolated and vaccines are formulated. The very first vaccines developed using recombinant DNA technique was Recombivax HB, which was developed by Maurice Hilleman in the mid 1980s, which was approved by FDA in 86. Other vaccines produced using recombinant DNA techniques can be seen in the following table, where you have the brand name and then the disease in the second column and the particular antigen which is the main vaccine part, and the various expression system that is used for production of these vaccines. So, let us just go through one or two examples. For example, these hepatitis B vaccine, the brand name known as Angerix B, the particular antigen gene that was cloned is HBsAg, and it was expressed in Saccharomyces cerevisiae. Similarly, there is a SARS-CoV-2 vaccine which is sold by the brand name Nuvaxovid, and the SARS-CoV-2 spike 2 protein gene was cloned into baculovirus insect expression system. So, similarly, there are others like Gardasil cervarix. So, kindly please go through this table for a full update. So, let us now discuss about DNA vaccines. These vaccines deliver antigen coding DNA sequences into an organism's cells to stimulate an immune response. A genetically engineered plasmid containing the DNA sequence for the desired antigen is injected, and once inside the cells, the DNA is translated into the antigen, triggering immunity. These vaccines can stimulate a broader range of immune response compared to traditional vaccine. In August 2021, Zykov D, developed by India's Cadila Healthcare, became the first DNA vaccine to receive emergency approval for human use. Now we have these principle illustrated over here. So, DNA vaccines mimic natural infection to stimulate an immune response. Once inside the host cells, the DNA is transcribed and translated to produce antigens. These antigens can be displayed on the surface of transfected cells by MSC class 1 and class 2 molecules. Here you can see the site of inoculation once the vaccine is formulated using rDNA having the gene of interest. So, these are getting presented into the antigen presenting cells, or it can be secreted by transfected myocytes and later processed by dendritic cells, as in the case of exogenous antigens. APCs then travel to lymph nodes where they activate T cells, initiating a robust immune response. DNA vaccines consist of plasmid with a strong viral promoter such as SV40 promoter, RSV promoter and CFB promoter. Polyadenylation transcriptional termination signal sequences as you can see over here from bovine growth hormone or rabbit beta globin improves the mRNA stability. Many DNA vaccines use polycystinic vectors for the expression of more than one immunogen. Other components are enhancer sequences, synthetic introns and adenovirus tripartite leader sequences which improve the expression rates. Then there is a N-terminal ubiquitin signal which is added to make the express antigens liable to cytoplasmic degradation. Let us now discuss about the RNA vaccines. An mRNA vaccine utilizes a replicated form of messenger RNA to initiate an immune response. This mRNA once inside the body serves as a blueprint for constructing a foreign protein which then trigger an adaptive immune response. So, the delivery of mRNA is facilitated by a co-formulation that encapsulates the RNA in the lipid nanoparticles and aiding their absorption into the cells, as you can see here in this picture. The advantage of mRNA vaccine include ease of design, rapid production, cost effectiveness and induction of both cellular and humoral immunity. So, this viral antigen mRNA is actually transcribed and translated and the viral antigen is produced which then interacts with the class 1 and class 2 MSCs leading to both cellular and humoral immunity. Then these mRNA vaccines are produced by the in vitro transcription of plasmid DNA templates containing an RNA polymerase promoter and a target gene sequence using T7 phase RNA polymerase. The synthetic mRNA mirrors naturally eukaryotic mRNA with key structural components, a 5' cap, 5' UTR or untranslated region. Then you have the 3' UTR here, an open reading frame and which encodes the antigen in a 3' poly A tail. These components can enhance mRNA stability, translation efficiency and vaccine efficacy within the target cells. So, this is the carrier vector and this is the vaccine under after formulation. Let us now discuss how these vectors are delivered. One of the way to deliver is using peptides as a delivery agent. Peptides, particularly those containing positively charged amino acids like lysine and arginine, form electrostatic complexes with negatively charged mRNA. Protamines are especially advantageous as carriers, protecting mRNA from RNase degradation and enhancing immunogenicity through structural similarity to viral RNA. However, despite their high adjuvant activity, protamine mRNA complexes have shown poor translation efficiency. So, there are other systems which are available like the polymer based delivery systems. Polymers are effective materials for mRNA vaccine delivery. offering protection against RNAs. To improve stability, polymer-based mRNA nanoparticles are modified with lipid chains, hyperbranch groups or biodegradable units. Common cationic polymers include polyethylamine, polyamidoamine, dendrimers and chitosans. Then we have the lipid nanoparticles, which are widely used for mRNA vaccine delivery, as they can protect mRNA from RNAs and can facilitate intracellular delivery via endocytosis. Lipid nanoparticles are composed of synthetic lipid materials, including cationic or ionizable lipids such as DOTMA, DLIN-DMA and TT3. Moreover, cyclic amino head groups can enhance immune response by activating the STING pathway. Then we have the cationic nanoemulsions or CNEs, which utilizes nanoemulsions with cationic lipids for mRNA delivery. These nanoemulsions stabilize an oil core in an aqueous phase using hydrophobic and hydrophilic surfactants and are produced through methods like agitation, ultrasound and microfluidics. MF59 on oil in water nanoemulsion is an adjuvant used with flu vaccines for the elderly. Then we have the viral replicon particles. These are used to deliver self-amplifying mRNA into the cytoplasm, mimicking viral infection. This allows efficient mRNA replication and antigen expression, leveraging the high efficiency of viral internalization and genome release. Then we have the common VRPs, which include single-stranded RNA viruses like alpha virus, flavivirus, rhabdovirus and measles virus. So, here in this table, list of vaccines which are approved against certain diseases listed in column 1, like for example, SARS-CoV-2, and the vaccine name like the Spikevax, or it is also alternatively Soles Comirnaty, and produced by two different companies, Moderna the first one and Pfizer the second one, and these are currently have received approval, and the formulation they use is lipid nanoparticle. So, we can see that lipid nanoparticles has been used extensively in delivering various vaccines against various diseases like rabies, influenza, respiratory syncytial virus and so on, in various phases of clinical trials, some in clinical trial 1, some in 3 and some in 2, and some are already approved. So, we can see that formulation is an important part of this vaccine development process. So, in 2023, the Nobel Prize in Physiology or Medicine was awarded jointly to Katalin Kariko and Drew Weissman for their discoveries concerning nucleoside base and modifications that enable the development of effective mRNA vaccines against COVID-19. So, now viral vectors are also used as a vaccine. A viral vector vaccine uses a modified virus to transport genetic material that, when transcribed by the recipient cells, generates mRNA encoding a specific protein or antigen prompting an immune response. Unlike subunit vaccines, viral vector vaccines activate cytotoxic T cells, generating a robust immune response beyond solely humoral immunity. These vaccines leverage a modified virus as a delivery vehicle for nucleic acids encoding particular proteins, aiding in immunity against the disease and protecting against infection. Some of the viruses which are used in viral vector vaccines are adenoviruses. These are popular in vaccine development for their high transduction efficiency, strong transient expression and broad tropism. Most vectors used are replication defective vectors via deletion of the E1A and E1B genes to enhance safety. As of April 2021, 4 adeno-based COVID-19 vaccines were authorized. Then we have the adeno-associated viruses from the Parvoviride family. These are non-enveloped viruses with a 4.8 kb single-stranded DNA genome. These vectors are popular for gene therapy, as these vectors possesses inverted terminal repeats on both ends of their DNA, which enable them to integrate their genetic material into the host genome, thereby modifying the host genome. Then we have vesicular stomatitis virus, which is a member of the Rhabdoviridae family. It is bullet-shaped, single-stranded, negative sense RNA virus. It is a valuable vaccine vector due to its ability to replicate at high titers, low seroprevalence and minimal pre-existing immunity in humans. Then another type of virus is the pox viruses, which include vaccinia virus and variola virus. These are large envelope viruses with a double-stranded DNA genome. They replicate in the cytoplasm using viral polymerases, a unique feature that enables their use as vaccine vectors. They can carry large genes up to 25 kb, making them ideal for multi-antigen vaccines targeting multiple pathogens. So, here we can see various vectors listed in column number 1 for carrying various vaccine candidates, and mostly here we see the extensive work on SARS-CoV-2, because this was one of the most dangerous disease in recent times. And then you find in this column the targeted antigen, particularly the spike protein in case of SARS-CoV-2. But then you have for Japanese encephalitis and dengue the viral envelope PRM and E. Then for the Ebola we have the glycoproteins and so on, and here in this list are the companies which have developed these different vaccines. So, with this we finally come to the end of today's lecture. Thank you for your kind attention.